Hello, my wonderful students, and welcome back. I know, exams are right around the corner, and this biotechnology chapter can feel a little complicated. But I promise you, it is one of the most exciting topics, and today we're going to make it super easy.
So, are you ready? Just relax. Remember my simple rule. No fear when your teacher is here. We are going to break down every single concept into tiny, easy-to-remember pieces. Trust me, by the end of this session, you'll be able to answer any question. Cello, let's crack this chapter together.
Okay, here's our plan for today. It's a simple little roadmap. We'll start with what biotech even is, then we'll look at the scientist toolkit. After that, it's time for the main recipe to make special DNA, see how to make billions of copies of it, and then we'll finish with a very important exam question. Simple, right? Let's begin.
So first things first, what in the world is biotechnology? You probably think of some high-tech labs with scientists in white coats, and, well, you're not wrong. But you know what? It's also something that happens right in your own kitchen. It's a massive field, stretching all the way from making dahi to creating the latest DNA vaccines.
Now, this is the official definition from the European Federation of Biotechnology. I know, it sounds a bit scary. Integration of natural science and organisms. Phew, but let's simplify it. All it really means is using living things, or even just parts of them, to make stuff that's useful for us humans. That's it. See? Not so scary.
So, making curd, or bread, or wine, that's what we call traditional biotechnology. It's been happening for centuries. It just uses whole organisms, like bacteria or yeast, pretty much as they are. But what we're really focusing on today is modern biotechnology. This is where we get into the cool stuff, like genetic engineering, where we actually change an organism's DNA to get exactly what we want. Things like making vaccines or even fixing broken genes.
Okay, so if we're going to be genetic engineers and start playing with DNA, what do we need? You can't just go in with your bare hands, right? Just like a carpenter needs a hammer and saw, a biotechnologist needs a very special toolkit. So let's open it up and see what's inside. Inside our toolkit, there are four main things. First, we need special scissors to cut DNA. These are our restriction enzymes. Then, we need some glue to stick the DNA pieces back together. That's DNA ligase. We also need a delivery truck to carry our DNA into a cell. That's the cloning vector. And finally, we need a factory where all the magic happens. That's our competent host. Scissors, glue, delivery agent, and a factory. Got it?
Now let's zoom in on the first two tools, which are maybe the most important of all, the scissors and the glue. I mean, without these, we can't do anything. They're what let us do this amazing cut-and-paste job on DNA so precisely.
All right, let's talk about our molecular kainchi, our scissors. These are restriction enzymes, and a very famous one is called EcoRI. Now, these scissors are incredibly specific. They don't just cut anywhere, oh no. They search for a very specific sequence on the DNA called a palindromic sequence. You know, like the word Malayalam, it reads the same forwards and backwards. The enzyme finds that exact spot and snip. It makes a clean cut, leaving these things called sticky ends.
So, once we've cut out our gene and it has those sticky ends, we need to paste it somewhere else, don't we? And for that, we use our molecular glue, DNA ligase. Just think of it as the Fevicol of the DNA world. It perfectly joins the sticky ends of our gene to the sticky ends of our vector DNA. And voila, we've just created something amazing called recombined DNA.
Great! So we have the tools, we know the basic idea. Now for the main event, the actual recipe for making recombinant DNA. Now listen carefully, my dears, because this process is a very common exam question. You need to know these steps by heart. The recipe has 6 straightforward steps. 1. Get the DNA out of the cell. 2. Cut it up with our scissors. 3. Find and separate the one piece we want. 4. Glue it into our delivery vector. 5. Put that vector into our host cell. And 6. Grow that host cell so it can make our product. Now let's look at a few of these steps a little more closely. So, step 1 is getting our hands on the pure DNA. We have to break open the cell and get rid of all the other junk like RNA and proteins. Once it's all clean, we add some chilled ethanol, and it's like magic. The pure DNA suddenly appears as these fine white threads. You can actually see it with your own eyes, just like in this picture.
Okay, so we've cut our DNA, but now we have a whole mix of different sized pieces. How on earth do we find the one gene we're looking for? Well, we use a technique called gel electrophoresis. It's basically a race for DNA fragments. We put the mix in a gel and run an electric current through it. Since DNA is negatively charged, all the pieces move towards the positive side. But here's the trick. The smaller the piece, the faster and farther it runs, so we can see the bands and just cut out the one we need.
Now we need to get our gene into the factory, our host cell. And for that, we need our delivery agent, the vector. These are usually plasmids, which are just small circles of DNA that bacteria have. Now, for your exams, you absolutely must remember this famous one, PBR 322. See the diagram? It has some really important features. ORI is the origin of replication. That's where it starts making copies. And AMP-R and TET-R are genes for antibiotic resistance. These act as little selection markers that help us find the cells that have actually accepted our new gene. Very, very important.
Fantastic! So we've made our recombinant DNA and put it inside a host cell. But think about it. Is one single cell enough to make medicine for lakhs of people? No way! We need billions and billions of copies. So, how do you make crores of copies of a single gene and do it super fast? You use an amazing high-tech photocopy machine for DNA. It's called PCR, which stands for Polymerase Chain Reaction. This invention was a complete game changer, because it lets us take one tiny bit of DNA and make enough of it to actually see and work with. PCR is basically a cycle of 3 simple steps that we just repeat again and again. First, denaturation. We heat up the DNA and the 2 strands separate. Second, annealing. We cool it down a bit so tiny little starter pieces called primers can attach. And third, extension. A special enzyme called Taq polymerase comes and copies the rest of the strand. And the amazing thing about this enzyme is that it can work at really high temperatures because it comes from bacteria that live in hot springs.
And the power of PCR is just mind-boggling. Just think about this. By repeating that 3-step cycle only about 30 times, which takes maybe a couple of hours, you can get over 1 billion copies from just one starting molecule of DNA. 1 billion. That's more than 100 crores of copies. Just incredible.
Right, so PCR gives us billions of copies of our gene, which is the recipe. But we don't just want the recipe, we want the final dish, right? We want the protein. For that, we grow our host cells. And to make huge quantities, like in a factory that makes medicine, we can't just use small lab equipment. We have to scale up to these massive, sterile steel containers called bioreactors. These are like giant, perfectly controlled pressure cookers that provide the perfect environment for our host cells to grow and produce our protein in bulk. All right, my champions, you have learned the entire process from start to finish. You're doing great. Now, let's put it all together and practice with a very important question. This type of question comes up a lot in the board exams, so are you ready? Let's focus. Okay, here's the question. Think carefully. Why is the bacterium Agrobacterium tumefaciens called a natural genetic engineer? Think about what it does in nature and how we've used that. The answer is so clever. So this bacterium is basically nature's own expert hacker. In the wild, it infects plants and injects a piece of its own DNA, called tDNA, into the plant's cells. And this causes the plant to grow tumors. Now, some brilliant scientists saw this and had a light bulb moment. They realized this bacterium has a perfect ready-made system for delivering DNA into a plant cell. So they took this bacterium and they disarmed it. They removed the nasty genes that cause tumors, but kept the beautiful delivery machine intact. And now we use this modified bacterium as a perfect little courier to deliver useful genes into plants. Absolutely brilliant.
So you see, the big idea here is that we are learning from nature and using its own incredible tools to build a better future. Like one of the fathers of this field, Herbert Boyer, said, "The breakthroughs we make today will become the everyday tools of tomorrow." So now I have a question for you. What's next? How do you think this amazing technology will change the food we eat or the medicines we take? Think about it. Okay, my dears, that's all for this revision. Study well, be confident, and you will do great in your exams. All the very best.